Hello, everybody. Attendees are coming in. Welcome to our 17th webinar with the Lara Perin Lab. Tonight, our host is Patrick, very conditioned in what he's doing, so I'm sure it'll be fabulous as always. Good luck to everybody this evening. I'm sure we are in for an interesting evening. The PowerPoints will be fantastic. If you have any questions, please put them in questions and answers. Patrick, over to you.
Thank you so much, Katie. It's fantastic to be back after the operation, and I'm really looking forward to some fantastic presentations. This is our 17th online scientific workshop. Today, we're extremely privileged to bring you 5 speakers, all from Laura Perin's Los Angeles lab. They will be discussing their research into Alport syndrome and, of course, answering your questions as well. For those of you who don't know me, I'm Patrick, and I'm a volunteer with Alport UK. I'll be hosting the workshop today. Let's introduce our 5 speakers, though. Firstly, Assistant Professor Laura Perin, who is the co-director of the GOFARR Laboratory at Southern Research Institute at Children's Hospital, Los Angeles, at the Keck School of Medicine of USC. Dr. Perin is trying to understand the molecular mechanisms responsible for glomerular damage, and she is also working on the development of potential new therapeutics for chronic kidney disease. Next, Dr. Camille Nicholas Frank, who studies how the cell cycle influences podocyte biology and survival during the progression of Alport syndrome. Thirdly, Charmi Dedhia. She is a research specialist who studies the role of MIR93 in disease progression in Alport syndrome. She is also studying the potential therapeutic role of extracellular vesicles in repairing glomerular damage. Fourthly, Dr. Hasmik Solayan. Dr. Solayan focuses on studying the role of glomerular endothelial cells, or GECs, in glomerular damage, with a specific focus on Alport syndrome, which is lucky, otherwise we wouldn't have her here today. Finally, Dr. Astgik Solayan, who has been working on the development of a glomerulus on a chip to study glomerular filtration barrier biology. Finally, our scientific moderators, who will be helping me pose scientific questions to our wonderful speakers. Firstly, Dr. Richard Naylor. He is a research associate in the Lennon Laboratory at the University of Manchester, specialising in disease models for Alport syndrome. And for the first time, Tomohiko Yamamura of Japan, who is currently working at the Lennon Lab, researching exon skipping therapy. Right, I think that's quite enough from me. It's time to kick off with our first presenter, Laura, to introduce the lab and give us a brief overview of their research. Laura, I can see, is already sharing her screen, so would you mind taking us away?
Good evening to everyone. I really want to thank the UK Alport Foundation for inviting us tonight. It's an extremely great honor for me and my lab to be here and show you some of the work that we do on Alport syndrome. We are under the Division of Urology at Children's Hospital, and we are the GOFARR lab. So you can see here on the left, this is the Children's Hospital, and on the right, our research building, the Southern Research Institute, and we are located at the 5th floor. This is a professional picture, as we used to do in LA, but really this is how we look like in the lab, and particularly on the left are the people that are now in this year working in the lab, and you will hear from them the following presentation. So as I said before, we are the GOFARR lab, and I don't know if many of you knows, but GOFARR stands for Gabrial Organization for Renal Research, and we are here thanks to the Schenker family. So Michael and Elisa supported our laboratory since 2006, and now thanks to them, we are the NIH-funded lab. Michael and Elisa Schenkman, like most of people that will listen to this talk, have a very important personal story. They had a son, Gabrial, which was born with a hypoplastic kidney. And in 2016, Elisa donated a kidney to his son, and now they are both healthy and they have a normal life. The Schenker family is not only helping us in the lab, but they have a very important role also in the Department of Nephrology at Children's Hospital. They renovated the nephrology department and they developed different programs. And lately, They have developed a very fascinating program on virtual reality for the kids that are in dialysis. And this makes the kids, of course, very happy when they stay in the hospital. So we are very thankful that we know this family, and they are our daily inspiration for our work, together with all the patients that have a chronic kidney disease. So what do we study at the GOFARR lab? We study chronic kidney disease. Everyone knows the high clinical relevance of this disease, and more than 30 million of Americans have chronic kidney disease. And it really doesn't matter what kind of chronic kidney disease you have. In the end, you will lose kidney function, and you will have a formation of sclerosis and fibrotic tissue in your filtration unit. So what we do is try to understand the mechanism that regulates this glomerular damage in our kidney. So I'm sure that many people that are listening to this talk and this workshop knows what a kidney is and what are the function of a kidney and this major structure of the kidney. But I think that it is important for me to give you a little bit of background so I can really set the background for the talks that the presenter will be doing after my talk. So the glomerulus together with the tubules, so this is the glomerulus, and then you can see the tubules here, together they form the nephron, which is the filtration unit of our kidney. And it's very important to know that no nephrons can be generated in adult life. We are born with a determined numbers of nephron, which in the humans is around 1 million. And if for any reason you lose the function of this nephron, or you lose a number of this nephron, they cannot regenerate. And so therefore, this is the main reason why you develop chronic kidney disease, and in the end, you will need a transplant. So in the laboratory, we are focused on the glomerulus, not so much in the tubular part of the nephron. And this is a picture that Astgik published in 2015. It's a picture taken with a very particular technology, which is the scanning electron microscopy. And you can see here in a three-dimensional structures how the human glomerulus look like. So this is a very fascinating structures in my opinion. This one here in this video that it's taken using another technology that we have at USC, particularly is a very good friend of mine, Dr. Patty Peterdis. This is a live imaging of, it's a live imaging of a mouse glomeruli. You can see very well now here how an alive glomeruli in a mouse look like. You can see that there are like a gray color, which is the albumin that is injected. Albumin is a protein that is filtered on the kidney, and this allows us to visualize the glomerulus alive in a mouse. So in particularly, I was always very fascinated, not only of the glomeruli, but a very particular structure that is within the glomeruli, which is the glomerular filtration barrier. It's technically a simple structure because it's only formed by two types of cells and a thin layer of membrane, but indeed it's a very complicated and very fine tuned regulated structures, which is responsible for the filtration activity of our kidney. One of the cells that constitute the filtration barrier is actually the podocyte. Podocyte is a very unique cell, has a typical characteristic of what is defined as epithelial cells, but its location and architecture and even function is extremely singular. The podocyte have food processes that interdigitate between them, which are the cytoplasmatic extraction of these cells, and they are responsible for the filtration. So it's one of the major cell type in our kidney that really determine what protein should be filtered and which protein should be retained in our body. One important characteristic of these cells is that multiple study has demonstrated that these cells are what we call post-mitotic cells. So there are cells that do not replicate. So again, you're born with a specific number of podocytes, and if you lose them, you need to find a way to replace them, or you need to find a way to stimulate their to prevent that they are lost in the urine. And this is one of the projects that we do in the lab. So Camille, the first talk, will actually explain to you a mouse model that she has generated to study the proliferation of these cells. Then we have the second cell type in the glomerular filtration barrier, which are the glomerular endothelial cells. We call them JAK. It's kind of a lot easy and short. And also, this is a very particular cell type, as much as the podocytes are the cells that form the capillaries within the glomeruli. And these endothelial cells, compared to all different types of endothelial cells that we have in the body, they have a particular fenestration, and also these fenestration are important for the filtration. And this is a work that we study a lot, these cells too, and this is a project that Hasmik is going to talk to you later on, and she also developed a mouse model of Alport syndrome to study particularly only endothelial cells. So the podocytes and the endothelial cells are separated between them. There is a thin layer of glomerular basal membrane. This membrane is very unique too, and we know we can say that it's the culprit of Alport syndrome. This thin layer or matrix is composed by different type of proteins, but one of the major components is the collagen 4, alpha 3, 4, and 5 that indeed is altered in Alport patients in all our animal models that we study in the laboratory. Another important concept to keep in mind is that only podocytes are secreting this type of matrix within the glomerular filtration barrier. So they are responsible for the secretion of this matrix. I'm sure that you all know very well what Alport is, but specifically, as I already said, these podocytes in Alport patients are not capable of producing the proper collagen 4, alpha 3, 4, and 5, but it's a retain in the adult life, the collagen 4, alpha 1, and alpha 2, which is a more an embryonic form of the collagen, which is not capable to really allow the proper filtration to occur. And you can see here on the bottom the imaging where this thin layer of membrane is not anymore thin like a normal membrane, but is actually abnormally split and is laminated. So you can imagine that if a podocytes sit on this not properly constructed membrane, they're actually what we call not happy at all. They detach, and over time you lose your filtration capability. The clinical manifestation of Alport syndrome are known from my audience for sure more than me, which I'm not a nephrologist, but I am a basic scientist. But patients that have Alport, they present proteinuria and progressive kidney failure, and also they have a secondary effect like a hearing loss of ocular manifestation. This is a video, and I think the videos usually are very strong. They give a lot of more information than a lot of different graphs and science approach, because if you remember before, I show you the video of an intravitreal, again, a live imaging of the glomerulus here. You can see, a very organized and a very well structures of the mouse glomeruli. But if you image a live image of a glomerulus of a Alport mice, and this is a glomerulus of a mouse, more or less, I would say 5 to 6 months of age, where it's almost on a, in a we can say four degree of chronic kidney disease stage in the human, you can see very well that the capillaries within the glomerulus are not properly formed. There is overexpression, and you can see in green of alteration of glomerulus protein, and you can also see around the glomeruli here in blue the deposition of fibrotic tissue. So there is a very big difference just if you look at a live imaging of a glomerulus of a healthy mouse compared to an Alport mouse. So what do we do in the laboratory to study Alport syndrome, and what are our major projects? So in the laboratory, I'm not the only PI, I'm the senior PI, but Stefano and Sarkis now, they have their own line of research. They were postdoc in my laboratory. Now they are independent, they have their own funds, and they do their project, but we are under the same umbrella and we work together a lot, especially in the Alport syndrome, because I think that all three of us have Alport syndrome as one of our major passion for many years right now. So one of the major projects I have personally in my laboratory is the search cycle, as I've told you before, and you will hear from Camille about that project. Then I'm working very close with the Sarkis and Charmi on the roles of mirs. Mirs are a small sequence of RNAs, which is known lately in the few years, recent 5 or 6 years. These little tiny sequences are short sequences of RNA that have an incredible role in the progression of Alport syndrome and also in many other diseases. So we are studying specifically 2 mirs, which they have a completely different function in Alport, and you will hear from Astgik and Charmi. One is upregulated and one is downregulated. And then also Charmi is working with the extracellular vesicle, which we have shown and published that they have the capability of slowing down Alport syndrome progression. And so we think that this can be in the future with further characterization and more study, a possibility. They can be used as a possible therapeutic approach to slowing down progressive disease in Alport syndrome. Then you will hear from Hasmik and the role of the glomerular endothelial cells in Alport. And this is a project that Sarkis is developing. He was very interested in working with these cells. So Sarkis is more focusing on the glomerular endothelial cells, while I'm really focused on the podocytes. Then you will hear from Astgik, and this is a project that Stefano and I started a long time ago. We use a particular plate where we are trying to recreate in vitro what we call Alport glomerulus on a dish. We have published about this technology, and we are now really happy to keep working in this project because it's really a nice model that we can use to understand mechanism of disease progression in Alport. Then with Sarkis, I also have another project always in Alport syndrome. We are developing and studying a lot lately. We're using a spatial transcriptomics, which is a particular type of sequencing where we can study gene expression of the cells, specifically in the Alport glomeruli, but we can also determine where this gene expression is located in the biopsy of the Alport sample. And you will hear some of the results in Hasmik talk about this spatial transcriptomics. And I think it's very powerful because we can not only determine the change of gene expression that is between normal glomerulus of Alport, but also where we can actually see under the microscope where this gene expression and in which cells this gene expression is altered. But We are not only an Alport lab, we work in the kidney, but Stefano is developing also another new line of research where he's focusing specifically in studying membranous nephropathy, which is another type of chronic kidney disease. And I think that for us it's very important to study different type of kidney diseases so we can really determine if a different mechanism are similar to all chronic kidney disease, and possibly we can develop a therapy that work for not only Alport, but maybe also other disease like membranous nephropathy. Then together with Valentina, we do a project that is very dear to me, and I'm very passionate about this project because we are having very good results. We are developing a dietary intervention for chronic kidney disease. We have now done small clinical trials with patients with CKD using this particular diet. We have also used this diet in different animal models, and we have greater results because we can see that there is an amelioration of the kidney function when patients or our animal models are using this regimen. We are now moving towards to use this dietary regimen in Alport syndrome too, in our animal model. And I'm very hopeful that it will be positive too. And it's known that variation of dietary regimens are important also and have great effect for other chronic kidney disease of a genetic nature, like for example, polycystic kidney disease. It's already published that there is a particular diet that has the capability of reducing the size of the cyst in polycystic kidney patient. So we are now publishing this paper, and if you have any questions, please feel free to contact me or Valentina. Another project that Valentina had, she was always passionate about pancreas, which is nothing to do with the kidney, but it's a very nice project too and very interesting. And Valentina, together with Astgik, is also studying renal development and pediatric renal cancer, and in particular Win's tumor. So we're very lucky that our laboratory, being very close to Children's Hospital of Los Angeles, we have access to a lot of sample of this renal cancer of pediatric origin, and we are trying to understand what are the mechanisms also of development of this cancer and how the kidney develop, because we think that if we can understand how human kidney develop, maybe we can gain some knowledge on how we can induce a regenerative pathway in the adult kidney when is damaged for any different reason. And then finally, we have Paola, which is a collaboration that I have with the University UCLA, which is not related to kidney or pancreas, but she's working on muscular regeneration. And she just published a paper where she showed the importance of the thyroid hormone in muscular regeneration. With this, I hope I give you a little bit of introduction, and I was able to set the tone for the other talks that will follow. Again, I want to thank for sure, the Schenkel family, all my collaborators, all my funding, but really, I think that the core of my laboratory, I really want to thank the Alport patient, because without their support and they're willing to donate their amniotic fluid, which is one of my major interest of source of cells, and you will hear from the talk, we will never be able to do this research. So I'm really thankful for the patients who really participated to my research and are happy to do it and are trying my dietary intervention. And with that, I think that Camille can start with the presentation unless there are any other questions.
I certainly don't have any. It was fantastic to hear that your lab, in a sense, is funded by patients as well. Not only are you having this massive contribution from Alport's patients, so it's fantastic to see examples like this of patient-scientific collaboration. Thank you so much, Laura. I agree. I think we should move on to Camille, if that's possible. Okay, fantastic. Sharing your screen. Go when ready. Thank you.
Hi, good evening, everyone. Thank you to give me the opportunity to talk about our work on the role of the cell cycle in Alport podocytes. So just to give a little bit of background, but Laura already mentioned it. So we are studying the podocytes that are post-mitotic cells, which mean that they do not replicate on a healthy condition, and we are born with a certain number of podocytes for all life. However, we have found that it has been demonstrated that there is a loss of podocytes during the development of chronic kidney disease. And actually, this podocyte loss itself is deleterious for the development of glomerulosclerosis. As they are not supposed to replicate, they don't activate their cell cycle when they are healthy. However, it has been demonstrated that in certain kind of injury or stress, we can find podocytes that have activated the cell cycle, as we find podocytes that have 2 nuclei and so are almost ready to divide themselves. And also those binucleate podocytes are also found in the urine of the patients. So our goal was to study specifically the cell cycle of podocytes using a new tool that has been developed the last decade, which is called the FUCCI system, about fluorescence ubiquitination cell cycle indicators. To summarize, it's based on 2 proteins that are coupled to a fluorochrome with a specific color, and which allow us very easily to determine and visualizing which phase of the cell cycle the cells are. And they are, as they go into the cell cycle, they become red to be in G1, and then they are a little bit yellow for the very narrow S phase, and then they become green when they are in G2 and M phase. And you can see here how the cells are changing color as they are entering their cell cycle. And so this FUCCI system has been applied to mouse model when we can actually visualize cells in vivo and determine their cell cycle. And what we wanted to do is to use this mice with the FUCCI indicators and to study the podocytes. To do so, we crossed those FUCCI mice with a mice that is able to specifically express those marker only under podocytes. So only the podocytes in this mouse is going to have those colors for the cell cycle. And first, we wanted to look at how is the distribution of cell cycles in a healthy mouse. And as we expected that most of our podocytes are not into the cell cycle, or at least in the very beginning, the G0 phase, they don't have any color. And using flow cytometry analysis and selecting those podocytes with a nephrin antibody, which is a protein that only is expressed in podocytes, we found and confirmed that most of our podocytes, 95 to 17% of podocytes are actually in G0. And about 2 to 5% of podocytes are in G1 expressing red signal, and few are in S and in G2. And here in this picture, you can see that once we extracted the glomeruli from those mice and plated in vitro, they activate their cycle. It is well known that when we put a cell in a plate, in a dish, they're going to activate their cell cycle because they're not anymore in an in vivo system. And we can see here that podocytes are then entering cell cycle expressing those different colors. Finally, we confirm that as podocytes enter the cell cycle, they're actually getting bigger. But so our goal was really to study this cell cycle in Alport syndrome, which is a very good mouse model to study chronic kidney disease. So we use the Alport syndrome with X-linked collagen 4A5 mutation that our laboratory is very familiar with, as it very correlates the development of the disease in mouse to the development of the disease in human. So we cross those mice Alport with our mouse with pod FUCCI to create this mouse with that is triple genic. And we confirm that this group of colony do develop the Alport disease as they start to develop a proteinuria between 2 to 4 months old and have a very high range proteinuria 6 months old, which is about the lifespan, and they also have an increase of serum creatinine, which reflects the development of the chronic kidney disease. And so we looked at different age of the disease, the activation of the cell cycle, and we can see very well here how the progression of the Alport chronic kidney disease is associated with activation of the cell cycle in podocytes. At 6 months old, when mice are actually at the end-stage renal disease level, about a third of the podocytes are in G1 phase. And we can see here again that as they enter the cell cycle, the podocytes are actually getting bigger. The other step we wanted to do is to see if this activation of the cell cycle is actually correlated to the severity of the disease. And to do so, we use the mice that are female and heterozygous for the Alport mutation, as we know that those female do develop a milder disease, which is also the case in the carrier women in human who present a milder disease. We can see here that those mouse female heterozygous at 6 months age have a proteinuria that is about a third of the male at the same age and still have a normal serum creatinine. However, we show that they actually already have sign of cell cycle activation about to 15%, which is about half of the male at the same age, but it is much more than the wild type female or wild type male. So we can see that the cell cycle progression is actually correlated with the severity of the disease, and that it actually starts before the renal function is impaired itself. The final part of our study was to analyze the protein expression in those different podocytes at the different phase of the cell cycle. So to do so, we isolate the podocyte from our wild type and Alport mouse, depending on the phase of the cell cycle, and perform proteomics with the collaboration of the Pacific Northwest Laboratory. Because the limitation of a number of cells, we could only study 3 groups of cells: podocytes in G0 in wild type, podocytes in G0 in Alport, and podocytes in G1 in Alport. And what we found is using a different group of protein expression, that those groups actually cluster within the same group, but also very well separately between groups. Here, using the principal component analysis 2, which is a whole group of protein, we can see that the podocytes in G0 are very separated from the Alport G0. And using the group in the PCM1 component analysis, we are able to differentiate very well the podocytes in Alport that are in G0 from those who are in G1. Then we study those, what type of protein are expressed in those different groups. And we can see here that podocytes in a wild type G0 do have a higher expression of protein implicated in the mitochondrial and energy pathway, whereas both of the podocytes in Alport have an increased expression of protein expressed in the extracellular space and related to collagen. Finally, between podocytes in G0 and in G1 in Alport, we found that the podocytes in G1 do have a less expression of protein related to the cytoskeleton actin. Then we wanted to study one group compared to the other, and when we compare the podocytes between wild type and Alport in G0, it confirmed, and we were able to actually isolate different kind of protein and pathway, especially in the metabolic pathway. And studying the podocyte between G1 and G0, we were also able to specifically target a certain type of protein. What is interesting is that the studies that has been done in mouse, we were wondering if it could be correlated to what we see in the human. And with this study, we were able to find that PDLM2, which is a protein that is specifically expressed in glomeruli and is implicated in the food processment of the podocytes, is upregulated in the podocytes in G1 in Alport mice. And we used a biopsy sample from 2 patients. One patient with a male 26 years old with Alport syndrome, and we can see here with the green that there is a higher expression of this protein, PDLM2, compared to a healthy control of a 59-year-old man. So we can confirm that what we find with this proteomic study can be correlated to human studies. So to conclude, we were able to create a new tool to study the podocyte cell cycle in vivo that we have applied to the study of the development of Alport syndrome. And we show that podocytes indeed do activate and enter the cell cycle as the disease progresses. And finally, we were able to target 2 different pathways, one in the metabolic and energetic pathway between wild type podocyte G0 and Alport, but also between the rearrangement of the cytoskeleton in podocytes in G1 in Alport. Now we want to go further and try to identify some molecules that could actually interact with this cell cycle and maybe be able to slow down this activation and slow down the progression of the disease. I want, of course, to thank all the members of the GOFARR group and especially Laura and Dr. Kevin Lindley.
Thank you very much, Camille, for an incredibly detailed talk and some fascinating research. I want to throw now to Richard, one of our scientific moderators, who I believe has some questions on the research. Richard.
Hi, thanks, Camille. That was really interesting. I think getting the FUCCI to work in those mice is a real advance technically and like really impressive work. I was wondering, why do you think the podocytes are getting bigger and undergoing hypertrophy? Is that just a consequence of them losing potentially their foot processes?
So our hypothesis is, as we have podocytes that are lost in the urine, some part of the glomerular basal membrane is uncovered, and as the remnant podocytes getting bigger, then it would be a way for them to compensate and try to like cover all the basal membrane that has been uncovered. And so it could be a good way to compensate. However, if they activate their cell cycle too far, then it could be also at the end deleterious, and the podocyte could also be lost in this way. But we think that initially it might be a compensatory mechanism.
Cool. So they're going back into division and to compensate for potentially the loss of podocytes in the disease in Alport syndrome. Is that, do you think, a consequence of the podocytes de-differentiating maybe back to like a more kind of embryonic like state as such, and therefore they're more prone to re-enter the cell cycle? Or do you think it genuinely is an injury response to try and replace the lost podocytes? I know it's a difficult question to answer, but I've always been interested as to why, you know, what is the reason for those podocytes doing that, and maybe any insight in that.
It's definitely a question that remains to be asked.
Yeah.
And also the question is, as the podocytes in Alport actually have a dysregulation of their cell cycle itself that make them actually not be able to cover and to like compensate properly compared to a healthy podocytes. But we believe that it's more like, initially it's a good mechanism probably, but that leads towards the end, unfortunately, to more damage.
Right, so it could be like a sort of a way to repair small bits of damage that might be something that's sort of normal physiology, but when it becomes really serious, it's pathophysiological. Okay. And just one final question was from me is, I may have missed this, but the PD-LIM2, what kind of functions has that got? Do you know? Is that
it's specifically it's part of the food processment and the slit diaphragm to like assure that they are like well maintained, that each food processes is like in good shape, and it's part of that mechanism.
So that goes up in the, was that right? That was going up in the Alport patient?
It goes up in the Alport, and our hypothesis would be, is it like podocytes are trying to like upregulate this production to try to like maintain as much as they could their food processes before, unfortunately, they are too damaged.
So do you think that's going to be maintained in other diseases, not just Alport syndrome, but maybe, you know, membranous or FSGS or things like that?
Yeah, maybe Laura, you know, I don't know about other diseases.
Wait, maybe you'll look at that in the future, maybe.
Yeah, it's actually known, for example, in focal segmental sclerosis, that this is altered too, because it's one of the most important regulators of the actin cytoskeleton. So probably is also present in other type of disease.
Cool. So I just noticed, and there's a lot of questions in the Q&A. Sorry, I missed those, but I'll pass over to Tomohiko, and Tomohiko can ask.
Yeah, thank you for a nice talk. So there, we have several questions from anonymous attendee. So one is very important, maybe from the patient. So how do you identify therapeutics to impact the cell cycle? Yeah, it's a very, very important view. So yeah, or yeah, it's my own question is, yeah, is there any attempt to moderate cell cycle in other fields than kidney disease or something?
So it is a very interesting question. So the cell cycle, there are a lot of drugs actually that have been applied, especially in cancer, in oncology, because it's like used as chemotherapy. As you stop the proliferation cells of the cell cycle, then you're able to maybe stop the proliferation of cancer. And so one of our hypotheses is like we really would like to try one of those drugs to slow down the cell cycle of podocytes itself. However, the negative part of this is that usually those drugs are linked to a lot of side effects and can impact the whole all of the other cells and can be also very deleterious. So there is a very narrow path to study, but we definitely have some molecules that we would like to study.
Thank you. So, but we have many questions in the Q&A field. So, and other question from anonymous attendees are how does how does circadian rhythm impact the cell cycle? So yeah.
I wouldn't know how to answer that question. No, I don't know if you.
Thank you.
This is something that we are, the next step of our work will be probably the FUCCI, you know, the mice in the stabularium in the basement, they do have a regulation, have a circuit, you know, they have certain hours of time and light and dark, so they kind of have that. But I think that is our next step, but we really didn't yet investigate that part of it, but it's absolutely very important question to look it up.
Okay, thank you so much. So yes, last question is from Stephen Frank. Yeah, is it known that metabolic pathways are altered during cell cycle progression, or do you speculate that the metabolic changes are related to podocyte injury, defective filtration, and proteinuria?
Well, it is known that the cell cycle, the activation of the cell cycle is linked to a lot of modulation of the metabolic pathway in cells itself. And so that's why it is very interesting to have be able to see that that's what we also find in podocytes. And now we definitely need to go further to try to understand better those dysregulation. But yeah, the metabolic pathway are directly linked with cell cycle activation.
Okay, thank you so much. So yeah, yeah, all right, yeah, almost all questions are answered. Yeah, so could I ask you, Patrick? So could I ask you to take over the moderate? Yeah.
Sure. Thank you very much, everyone. I know there are some questions that haven't been answered. We've got 5 speakers, so I do want to move on, but I'm sure the speakers today would be delighted to talk about them on email or indeed when we address them later. Our next speaker, our 3rd speaker, is Charmi Dedhia, and I think we have her talk up now. So please take it away. Can't quite hear you yet, Charmi.
Can you hear me better?
Yes, there we go. Yes.
Sorry. Hi, I'm Charmi Dedhia, and I'll be speaking about miR-93 and extracellular vesicles, EVs, and their role in Alport syndrome. Okay. So my focus in the lab is on the extracellular vesicles, or EVs. So all cells in the body release different types of membrane microvesicles, and these EVs released from the cells are integral component for cell-to-cell communication and regeneration, homeostasis, etc. These EVs contain different cargoes like proteins, lipids, nucleic acids, for example, microRNAs. Previously, we have shown that the EVs isolated from amniotic fluid stem cells were able to slow down Alport syndrome progression. We also established that these EVs can protect glomerular structure and function by downregulating VEGF expression. So one injection of these EVs in the Alport mice were able to reduce the proteinuria, as you can see. VEGF signaling is important for glomerular homeostasis, and disturbance in this glomerular VEGF have been reported in Alport syndrome. In lab, we use the Alport syndrome mice, where we've shown that the VEGF is upregulated in the glomerulus of these Alport. And VEGF is also increased in these Alport mice. So we were curious to see what components from these EVs are able to do that. So when we characterize the EVs, we found out these EVs are 171 nanometer in mode and express the same surface markers as that of amniotic fluid stem cells. And when we did the proteomics and RNA-seq of these EVs, we figured that one of the microRNAs is MIR93, that is highest expressed in these EVs. We are particularly interested in MIR93 because one of the major targets of this microRNA 93 is VEGF. So we know that the microRNA 93 is an important regulator of VEGF expression, and it is essential for the maintenance of glomerular homeostasis. So we tested the expression of this MIR93 in Alport mice glomeruli and urine samples, and we figured that the MIR93 expression was highly decreased both in glomeruli and in urine samples of the Alport mice. So we moved on to the human Alport kidney tissue, and it's important to see that both the Alport patients, Alport kidney tissue from these patients showed decreased amount of MIR93. And not just in Alport, we also saw that in the FSGS patient, the MIR93 levels were highly decreased. So it's safe to say that not only in Alport, but all kinds of chronic kidney diseases might be important. In those chronic kidney diseases, MIR93 would be an important microRNA. Not only just in the kidney tissue, but also in the urine samples, the MIR93 expression was decreased. So alterations of this MIR93 was consistently correlated with the expression of VEGF. So we were curious to see what particular cell of the glomeruli was responsible for the difference in the MIR93 expression. So we isolated the different kinds of cells from glomeruli, and we figured that the glomerular endothelial cell at 5.5 months, when the mice are really sick, the MIR93 expression in the JAK were highly decreased, and in mesangial cells at 3.5 months, where the onset of the Alport syndrome starts, the mesangial showed higher expression of these microRNA. So possibly the JAK and mesangial cells might be playing an important role in regulating MIR93. So our hypothesis is based on the fact that the VEGF is high in Alport and MIR93 is low in Alport. These EV can directly or indirectly might be transferring MIR93 to glomerular cells and be able to regulate VEGF. Now, since the MIR93 downregulates VEGF, these human EVs might be able to reestablish the glomerular structure and function. So to further study if the human EVs actually regulate VEGF in vitro and in vivo, we studied the damaged JAK and tested them with the treatment of EVs, and we figured that these JAK, the VEGF value of the treated damaged JAK were normalized. When we treated the sick Alport mice with EVs, the VEGFR2 values were also restored as that of wild type. We also confirmed the MRI localization of iron-labeled human EVs in mice kidney cortex, and also in the in vitro, when we labeled these human EVs fluorescently, the mouse and human JAK were able to uptake these EVs. So we wanted to see, are these human EVs capable of restoring normal MYD93 expression and its targets, not only in JAK, but also in other glomerular cells. So we tested the EVs on the damaged JAK and pods, and we saw that the MYD93 levels were restored. Not only MYD93, but its possible targets, for example, integrin beta-1, fibronectin, WT1, the markers of JAK and pods were also restored when we treated the damaged JAK and pods with these human EVs. So our final study for our hypothesis is that, can these EVs modulate VEGF by transfer of MYD93 to glomerular cells? So we tested the damaged JAK with EVs and damaged JAK with MYD93 mimic, and we saw that there was no significant difference between the action of MYD93 mimic and the human EVs. So it's safe to say that the human EVs behave the same way as that of MYD93 mimic. And to be sure that this MYD93 are from the EVs are what is responsible for the reverse of VEGF, for the reverse of modulating VEGF, we developed cell line where we knocked out MYD93, and so we isolated the MYD93 knocked out EVs, and now our further studies are going to be used, these EVs to be used on damaged JAK to see if MYD93 is what is responsible from these EVs for modulating VEGF. In conclusion, I would like to say that alterations of VEGF in Alport glomeruli correlate with the expression levels of MYD93. VEGF is one of the major targets of MYD93. Human EVs contain MYD93 as the major microRNA cargo. Human EVs also are uptaken by glomerular cells, and these EVs can be transferring MYD93 directly or indirectly to glomerular cells to restore VEGF damage in Alport. In future, these human EVs might represent a potential new therapy to restore glomerular structure and function in Alport. Lastly, I would like to thank my PIs and my current lab members. Thank you.
Thank you so much, Charmi. A typical patient question, I guess, whenever we talk about therapeutics is this bomb that you dropped in the very final slide, which is that this could have potential for therapeutics. Is this where you want to take your research over the next couple of years in terms of looking deeper into this issue?
Definitely, yeah. It has a very translational potential, this project, and human EVs are already being used in treating so many other different diseases. And we have gone so far to understand what in particular these human EVs are responsible and how they can reverse or restore the damage in glomeruli. So for sure.
Sure, and I think it's probably worth saying that if it applies to other diseases, it's probably perhaps slightly easier to get funding or backup for this, because you're not simply focusing on one particular disease that this therapy applies to.
Yeah, that's where Lara comes in.
Yeah, I'm sure it's a problem for the managers, I guess. Fantastic. Tomohiko, we've got some questions in the Q&A section, but I want to throw to you first for any questions you might have for Charmi.
Yeah, thank you for excellent talk here. I'm very impressed with your talk. So yeah, I have one question. So yeah, regarding the microRNA in Alport syndrome, so already the inhibitor of microRNA 21 is applied for the treatment for Alport syndrome. So yeah, the other way around. So is it possible to directly increase the expression of microRNA 93 in the mouse or human? Or so is there any possibility to apply such a way to treat with Alport or other kidney disease?
Definitely microRNA 93 is responsible for restoring the damage. I don't want to say too much, but Laura, do you want to answer that?
Yeah, so I think that we do have a lot of work in human vesicle, and we characterize them deeply with proteomics and RNA sequencing and all the study we have done. And we think that there is a possibility to possibly, get, we know that the AVs are transferring their cargo to the cells, and MYD93 expression is increasing when these cells are in contact with the vesicle in our main in vitro study and in vivo study too. Now, if it's a direct mechanism because MYD93 is directly transferred, or if MYD93 is activating other mechanisms by which in the end there is an increase of MYD93 and then down regulation of VEGF, that is the answer that we are trying to figure it out right now, if it's a direct or indirect mechanism. But in our in vitro experiment, it's totally possible to increase the expression of this mirror in podocytes or in JAK or in mesangial cells that we work with.
Okay, thank you. So we have a couple of questions from the audience. So first one from Joito. Where is the MR93 found to isolate for treatment of the mice? So is this from the amniotic fluid stem cell, and can it be cultured to create increased supply? So the derived from.
So
yeah, Laura, go.
So the MYD93 is in our case is obtained from extracellular vesicle that we isolate from the amniotic fluid stem cells that we work with. So we can expand these cells as much as we want. And actually we have done a lot of study about these cells. And so we can have enough supplies or MYD93 vesicle to be possibly scaled up and delivered to patients. So if we envision a treatment for future patients, possibly, when we are trying to figure out how to deliver and how often to deliver this microvesicle, possibly we will think that some multiple injection of this vesicle to the patients might really over time to slow down the progression of disease. They can be possibly, we were thinking that since the effect is quite long in time, because with only one injection in the mice, we can see a very prolonged effect. In the patients, we can say that maybe twice a year or 3 times a year would be enough based on our calculations and talking to nephrologists and working with this regimen. We think that this could be a possible treatment in this regimen for the patient. Hope I answered the question.
Yeah, thank you so much. So Richard, so could you take over the handling question? So
yeah, sure. Thanks, Tomohiko. So Professor Neil Turner asks, delivery of the vesicles to podocytes over the lifetime of an Alport patient could be a challenge, ? So do you envisage, or do you agree with that, or do you envisage different delivery modes?
Yeah, this is a very good question, and I spoke a lot with a lot of nephrologists on how this can be reached. And I think that the way in my opinion, if you deliver them systemically, they will not reach the podocytes in the way that we are expecting to have an effect. So the best way would be deliver them through the renal artery, if that would be possible in the patient. And then, as I said, possibly 3 times a year and that in that way they will probably reach the podocytes and the endothelial cells where we know that we see them there in the mouse. Systemically, unfortunately, I think that they will be dispersed too much, that they will not reach the kidney and the right amount that we will hope for to have an effect.
Okay. Hans George Roth asks, how would you distinguish a human EV from an RNA virus particle? I guess I had a similar sort of thing, because I was thinking about lipid nanoparticles, which to me sound a bit similar to human EVs. What's the difference? What is it that's making human EVs distinguished from those like virus particles, etc.?
So we are very focused now on study MIR93 because it really comes up, to be one of the highest MIR expressed. But I think that vesicle has multiple effects and have a multiple cargo. Then I don't know if a virus would be having the same effect of a vesicle, which contain the same cargo that you have in the stem cells. So it's a very particular cargo that reflect the cells of origin with a particular regenerative protein and regenerative mirs inside that are stimulating important regenerative pathway in the cells where they are uptaken. So I don't know how I would really distinguish one and the other, and which would be better at the other. I just know that the vesicle are have this cargo, and it's very complicated cargo. We focus on MIR93, but it's a huge, there are 2,200 mirs. So I'm sure that when they are delivering their cargo, they have different effect. And I think that the effect of all the cargo is actually what make them having this positive effect. Then I don't know how I will distinguish between the vesicle and the virus particle.
Okay, cool. Patrick, I think that's the Q&A questions.
Fantastic, thank you, Richard. And as I said before, I think our experts will be answering them live in the Q&A chat a bit later. Next, we have Hasmik Solayan. Would you mind sharing your screen and taking us away? Fantastic.
Hello, everyone.
So, I'm sorry, we can't quite see your screen, that's all.
Do you see my screen now?
Not. Oh, I think it may be an issue with Zoom. 1 second. I can't. No, I can't see your screen. It's not showing for some reason.
What about now?
There we go. Yeah, we can see it.
Sorry about that.
No worries.
Hello, everyone. So I am very excited to share my work today, the role of glomerular endothelium in Alport syndrome. Let me make it bigger. Glomerular endothelial dysfunction plays a key role in the development of chronic disease, but its impact on Alport syndrome is not fully understood. JAK are unique cells because they have this intracellular fenestrate, in Latin meaning windows, which are only found in kidneys and a few other organs. So one of the changes that we detected in our colony of Alport syndrome mice was that they had fenestrations that were much larger in Alport syndrome compared to wild type. I should also mention that these observations were made shortly before onset of heavy proteinuria. And one of the first candidate molecule we immediately checked that we know is involved in JAK homeostasis was VEGF. And as you see, VEGF was highly elevated in the same time point. So we have evidence that these cells undergo some key changes at microscopic and at molecular level during Alport progression, and this set the stage to further explore the molecular changes in JAK. In this regard, we generated a tectome reporter Alport syndrome mice to be able to isolate JAK and study them. This allowed isolation of JAK with high purity by FUCCI, and on isolated JAK, we performed RNA sequencing, and as you can see, this principal component analysis Alport syndrome JAK are completely different from wild type JAK. And as you can see, only 20% genes in JAK in Alport syndrome mice were differentially expressed, and this is also reflected in the heat map shown here. So then we did co-enrichment analysis for JAK genes, and pathways associated with lipid metabolism were highly enriched. Some of the co-terms as shown in the table include fatty acid metabolic process, lipid transport, lipid localization. We also performed ingenuity pathway analysis, and which predicted similar outcomes. We also performed, as you can see here, are mitochondrial genes that are altered as well. And so then we looked into specific genes, and as you can see from these heat maps, many genes involved in the fatty acid metabolism were downregulated, whereas genes associated with cholesterol pathway were more highly expressed, indicating a potential disbalance in the lipid metabolism in Alport syndrome JAK versus healthy JAK. In addition, we found that PGC1 alpha was also highly downregulated in Alport syndrome JAK. PGC1 alpha is transcription co-activator and is involved in many metabolic regulations. And also, I would like to mention that is involved in mitochondrial biogenesis. So then we have the following question. Do these changes at the molecular level in the lipid metabolic pathway have an effect on the glomerular structure and function, and can they be reversed? So one of the gross indications that there is a lipid disbalance in glomes is the histologic evidence of accumulation of lipid droplets in glomeruli of Alport syndrome mice, as illustrated here. This is all red staining on frozen section. And then we sorted out the JAK and measured the PGC1 alpha at a protein level, and we saw the results similar to the RNA sequencing data. So our current hypothesis is that lipid metabolism is altered in Alport syndrome glomeruli, and glomerular endothelial cells in particular contributing to JAK dysfunction, which can be reversed by extracellular vesicles derived from amniotic fluid stem cells. And we know that EVs are rich with lipid metabolism related cargo, such as fatty acid synthase, ATP citrate synthase. And to test our hypothesis, we performed proof of concept in vitro experiments using primary human JAK. So first we silenced PGC1 alpha and then treated them with EVs. And for the 8 hours post-treatment, both FASN and PGC1 alpha were slightly rescued. So these studies are ongoing, and I hope I could share them with you at the next opportunity. And I think this is all very fascinating. However, we wanted to test and see if is there any evidence of lipid metabolic disbalance in patients with Alport disease. So to tackle these and many other questions, we performed spatial investigation of kidney biopsies from Alport patient. So we compared human Alport syndrome kidney biopsies with healthy sample. We sequenced the human whole transcriptome atlas for over 18,000 genes and using a nanostring GeoMX digital spatial profiling platform, we analyzed 8 Alport syndrome glomes for male patients with collagen 4 alpha mutation and 11 healthy glom. And here, this histology image to the right demonstrates how the region of interest, in this case, the glomes were carefully selected using a polygon selection tool. So just to give brief overview how this DSP platform works, the slides are first stained with oligoconjugated RNA detection probes, then our region of interest selections are made from an image of the slide, after which the oligos are UV cleaved from the RNA probes and are aspirated and transferred into a 96-well plate and read on the counter system. Visualization of these results using principal component analysis showed that Alport syndrome glomes are different from normal as expected. And as you can see in these modified Venn diagrams showing the top 200 most highly expressed genes in each glome, they have many that are common, but also a few that are unique to each glome. In contrast, when we look at the 200 least expressed transcripts, we see that there are only small number of genes that are in common, and that there are a large set of genes that are unique to individual glomes. And the advantage of this technique is that it allows to study individual glomes and compare their similarities and differences. This is, I think, therefore a powerful way to study disease heterogeneity and mechanisms specific to one glome. And then in this table, we have the most important finding, GO analysis from a single glomeruli, where the top 3 biological processes are associated with lipid metabolism. And lastly, I wanted to show these heat maps, which represent heterogeneity of podocytes, JAK, and mesangial cells, cell markers between different Alport syndrome glomes. So finally, again, I hope I was able to demonstrate the lipid metabolism might be an important target in Alport pathology, and that AFSCs-derived EVs have the potential to reverse this process to normal. And thank you to my mentors, Sarkis Setrakian and Laura Perin, and my lab members.
Thank you so much. I think it's fantastic to see research on a topic that I don't think has particularly been explored in these workshops, at least not in detail. Leave it to the patient to ask a very basic question. You're talking about metabolization of lipids here, and I was wondering if you'd seen how we see these processes at the molecular level manifesting as symptoms in the patient. Do we see perhaps changes in weight or in health of Alport patients as the kidney function degrades and as these processes become more upset?
This is an interesting question. I think this occurring at molecular level when the glomeruli is damaged, and I don't know. Do you have anything to say, Laura?
Can you repeat exactly the questions, because I didn't hear it?
That's fine. I'm so sorry. I'll speak up. I think it's interesting that we've seen evidence of these changes at the molecular level in these processes, and I was wondering if we see these changes at the molecular level reflected as symptoms in the patient and changes in weight or something, or I don't know, something reducible from the Alport syndrome itself.
I don't have an answer on that, but it's certainly actually things that I never thought about it. So that is actually a good question. I think that maybe can be checked in Alport patients when they go to their nephrologist, and I can check on the clinical folder of the patients that from which I have the biopsies that we did the spatial transcriptomics, and they had a different stage and different age. Maybe there are clinical notes about their weight and other things. It's extremely fascinating. I never thought about that.
Well, perhaps it's a basic question, but as an Alport patient, I think we're all very used to having our weight checked regularly. So I was wondering if there was a correlation there. But I would like to turn to Richard now to. This is fantastic, Richard. I'd love to turn to you. I'm sure you've got a few in the Q&A as well, but by all means, I'm sure you've got some questions too.
Yeah, sure. Thanks, Hasmik. That was really interesting. Neil Turner asks, can you distinguish secondary effects via podocytes from primary endothelial consequences of sitting on the wrong matrix?
So there are publications showing that lipid metabolism is altered in podocytes as well. So this is a complex thing, and this metabolic alteration might affect the matrix as well, for sure.
Yeah, because I was intrigued because from my understanding, you showed that cholesterol, one of the changes you found was cholesterol went up in the Alport and lipid metabolism went down. And having seen Camille's talk, cells are proliferating and the podocytes are proliferating. So would you expect that, because for me, lipid metabolism, like one of the things that they'll make is phospholipids, that you're going to make plasma membranes, and cells that are dividing will make more phospholipids, but you're seeing it's going down in the glomerular endothelial cells. Is that suggesting that the endothelial cells may not be responding in the same way to the podocytes?
Yeah, as you mentioned, the beta oxidation and fatty acid synthesis are downregulated in Alport syndrome, JAG, and in contrast, cholesterol metabolism is upregulated. This might be some kind of a compensatory mechanism, and as you said, I think endothelial cell might reflect to these changes differently as differently to podocytes.
Cool.
Not the same.
So I'll pass on to Tomohiko because there's one more question in the Q&A.
Okay, thank you. Yes, thank you. Yeah, thank you for wonderful talk. So the question from Luis. So, what factors could account for the heterogeneity of Alport glomeruli? So, are there any group of genes commonly variable? So yeah.
Yeah, heterogeneity. So as I mentioned, the glomeruli, each glomeruli is affected differently, and also tubules are affected differently in this inside the kidney of this same individual. And what was your question?
Yeah, are there any groups of genes commonly variable? Yeah.
Yeah, there are commonly variable genes there are genes that are common for different glomeruli, and there are genes that are very individual for that glomeruli in particular.
Cool, thank you. Then another question from Stephen Frank. So, as a follow-up to lipid metabolism difference between podocyte and endothelium, is it known whether fenestrated endothelium have differing lipid metabolism or pathways to non-fenestrated endothelium?
I didn't know. Can you repeat the question, please?
Sorry, yeah, so yeah, maybe so it's better to answer by typing later. Or, yeah, I think, yeah. Sorry. Yeah, so yeah, anyway, so yes, so Patrick, so.
Sure. Thank you so much, Tomohiko. Yeah, I think Tomohiko's having a few internet issues, but that's fine. We are going to conclude our workshop now with a final talk from Astgik. Would you mind sharing your screen? Fantastic. Looks like we can see it. Not quite full screen yet. Brilliant. Brilliant, take it away.
Hello, everyone. Thank you for having me. Today, I'm going to talk about our innovative Alport glomerulus on a chip. So within our lab, we use a commercially available organoplate, which is a 3-lane system, and in combination with podocytes and human glomerular endothelial cells, identified as GEC. And we place these cell types in lane C, and we allow them to interact with one another. And when they do, they are able to recreate the glomerular basement membrane, which is vital for proper filtration to occur. And we identify that indeed they deposit the important components of the glomerular basement membrane, that is the collagen 4 alpha 3, 4, 5 trimer, and also laminin alpha 5, as shown through immunofluorescent staining and also via Western blot analysis. Furthermore, within our lab, we have previously published, Dr. Desako has published, showing that we could isolate from amniotic fluid human podocyte progenitor cells. And once we isolate these cells, we could actually differentiate them in vitro, and they behave very similarly to normal healthy podocytes and also to iPS-derived podocytes. Thanks to the Alport patients who were kind enough to donate their amniotic fluid, we are able to, currently we have 5 different amniotic fluid lines that carry various mutations ranging from collagen 4 alpha 4 to collagen 4 alpha 5, from which we have derived podocyte progenitor cells, and we've differentiated in vitro. The way we derive them is via 3 surface markers, CD24, podocalexin, and OB-cadherin. Once we derive these cells, we culture them in vitro and we're able to differentiate them. And now since we have the cells, we could actually place them back into the chip system and compare the Alport podocytes versus healthy podocytes. And we show that indeed Alport podocytes, unlike healthy podocytes, are missing the important glomerular basement membrane component, which is the collagen 4 alpha trimers. In particular, in this staining shown above in D, we see the presence of collagen 4 alpha 4 in healthy podocytes. However, in Alport podocytes, this is missing. And also we could observe this with Western blot analysis as well. Next, since with our chip system, we could actually test functionality. We wanted to see if, because they are missing the important GBM component, are they able to filter properly? And we see, unlike healthy podocytes, which maintain their filtration up to 60 minutes when we place albumin, Alport podocyte actually began to leak. And this leakage is quite significant. And we know that the glomerular basement membrane defects are the culprit of Alport. And now, since we have the system, we can actually begin to study signaling pathways that may be affected during this disease progression. We know that miR-193A is very important for glomerular homeostasis. And from our Alport podocytes, we identified that miR-193A is highly expressed. And from literature, we know that when miR-193A is highly expressed, it actually causes damage to podocytes. In particular, it causes foot process effetment. And it also is involved in other forms of chronic kidney disease, such as FSGS. And we know the downstream targets of miR-193A are WT1 and osteopotin, which are altered. In our mouse model that Camille presented along with Charmi, we've identified that indeed in Alport mice, miR-193A is upregulated and its downstream targets are altered. In vitro, we show that our human Alport podocytes also have elevation of miR-193A compared to healthy podocytes. But importantly, we show that we could actually target miR-193A using antigomirs and bring down this expression level. Now that we have the mechanism and we show in vivo and in vitro, we could apply it to our chip system and see if this is actually what is occurring as well. So what we did is we generated glomerulus on a chip of healthy podocytes and Alport podocytes. And then we isolated these cells and we ran qPCR on them. And we show indeed, similarly to the in vivo mouse model and also in vitro model, we see within the chip that the downstream targets of miR-193A are actually altered. And we know that this is due to the podocytes because the glomerular endothelial cells do not express miR-193A. So we see the same trends. Now that since we have this functional system, we could actually begin to study the filtrate and see what they secrete. So what we've done is we've generated glomerulus on a chip of healthy podocytes and Alport podocytes, and we've collected the filtrate. And we've analyzed the proteins that come through. And we've identified 80 human highly expressed proteins that we know are directly coming from our cells because the media components that we are using are composed of fetal bovine serum. So we are not using any human serum. And then we've compared the two samples and we've identified that there are 17 specific proteins that are expressed by the Alport. And these are fibronectin, ceruloplasmin, plasminogen, thrombospondin, and also serpins. And we know that miRs can regulate serpins. And glomerular dysfunction in Alport is correlated with a dysregulation of serpins, specifically serpin A3. And since we have this chip system, we could actually begin to investigate if modifying the miR-193A expression, could we have better functionality? And also what other signalings are affected. As a proof of concept, what we've done is we've formed Alport glomerulus on a chip in comparison to healthy podocyte glomerulus on the chip. And we show that indeed, Alport glomerulus on the chip do secrete an elevated level of serpin A3. But importantly, when we inhibit, when we target miR-193A, we actually are seeing a down regulation of the serpin A3 and its expression to normal levels. So with this system, we show that the Alport glomerulus on the chip is an excellent system to validate our mouse data, to study Alport disease mechanisms, and to identify markers, biomarkers. And we could go back into what Hasmik just presented using digital spatial transcriptomic profiling of the healthy glomeruli versus the Alport glomeruli and identified differences between them. And we showed that indeed, compared to what we've identified within our chip system, those particular proteins are also differentially expressed. We see elevated level of thrombospondin, plasminogen, vetronectin, and seroplasm. So with that, I would like to thank my mentors, Dr. Perin, and also Dr. Desako, and also all the GOFARR members, and also to everyone who's here listening to our talk. And thank you also to the patients firstly.
Thank you so much, Astgik. A fascinating talk again, and really interesting to see a very visual demonstration of the way that Alport cells can fail. I want to throw, in the interest of time, I want to throw straight to Tomohiko to ask a few questions about the talk.
Yeah. Thank you for excellent talk. So yeah, it looks very, very nice in vitro system. So yeah, I have one question. So yeah, this system is really beautiful and can be applied for several ways. So could it be can it be used to the screen the effect of existing drugs or future potential drugs to the membrane?
Yes, that is an excellent question. And Dr. Perin also and Dr. Desako are working on that. And if you would like to contribute, Lara, to that, or just, oh, you're muted.
Yeah.
So your volume, I couldn't hear you very well, but you asked that if we use this chip for drug testing.
Yes.
Yes, we do have actually the majority of our work is on testing different type of drugs to see if we once we use that, the filtration bar, the chip doesn't leak anymore. And we have actually published that with a common drug that is used, for example, in membranous nephropathy, and we could see that the chip was not leaking when this drug were added to the chip. And definitely this is the best use of this chip, actually.
Yeah, thank you. Cool. So yeah, it's very promising, I think. Yeah, thank you. And one question from Mary, she said, in the chip filtrate, have you looked for lipids? lipid filtration, or?
That is definitely something we could look into. And Laura?
Yeah, there was the 17 protein that were very specific in the Alport chip were not related to the lipids.
Okay, thank you so much.
At least in our experimental model, but yeah.
Okay, thank you. So could I hand over Richard? So.
Yeah, thanks, Tomohiko. So just one more question quickly from Neil Turner. He says, another beautiful presentation, thank you. Your newly formed proteinuric on a chip glomeruli seem to be behaving like a rather mature human glomeruli, several years old at least before we see much proteinuria or sclerosis with many of the molecules you describe. So do you, I guess the question is, is that a surprise to you that you're seeing like sort of proteinuria happening in your glomerulus on a chip? So, I guess.
Yes, we are using Alport podocytes, mature podocytes, and during the interaction, they have to deposit the appropriate type of glomerular basement membrane, and they're just unable to deposit it properly. And we're using proper levels of albumin to test that is found within the human system. So if we modify, we probably will see less leakage, but the leakage is over time. So we're not seeing the leakage all at once. So this is a 60-minute time frame. So we add the albumin, and over time, we do see the leakage after we measure it at 60 minutes. So it is not right away when we add the albumin. So there is some filtration happening, but over time, you do see the high levels.
I've got sorry, Laura, these are patient-derived podocytes, are they? Or
yes.
You got them from patient? Okay, cool.
We use human podocytes.
Laura, you have a question or a response?
Yeah, I just want to say that I would not classify this like all the podocytes. I would just say that when you do this in vitro system, first you have to be sure that you have your filtration barrier formed so that you don't have natural leakage just because the barrier was not formed. So we always have our wells or controls, our chip where we know that there is no leakage, and then you can, based on the damage that you decided to do, so you use different concentration and you have a different type of leakage over time. So if, for example, you use a high level of glucose, you have a very high leakage pretty soon. If you use a low level of glucose, you have less leakage. In the case of Alport, you have visually a lot of albumin fluorescence that go through because our little system in vitro has holes because there is no glomerular basal membrane there. There is no collagen practically. So you can totally regulate the system though based on the amount of damage that you want to create.
Cool. That's really interesting. Patrick, I guess you want to close up?
Yeah, sure, Richard. Thank you. Thank you, everyone. It wouldn't be an Alport UK workshop if we didn't ask for a little bit of feedback. And today that's going to come in the form of a word cloud. Most of you will be quite familiar with that. There should be a link coming up in chat quite soon. There we go. If you would just go onto that link and give us a word or two about how you're feeling today. We're going to share your responses on our screen, and you'll be able to see what everyone else has been feeling, learning about Laura Pennerin's lab's work. I would put something, but I'm adding 2 screens here, so for now I'll just watch it like all of you. Inspiring, fantastic. Give it a few more seconds to roll in. Thank you so much, everyone, for coming today. It's fantastic to see so much appetite for talks like this. Intrigued. I love chipper. It's so British, especially on such an international workshop. Hopeful, perhaps typed a little bit quickly. While I'm letting that roll in, I just want to plug our remaining workshops. So we have 2 coming up that we want to highlight. Our workshops this year will feature the latest research from Alessandra Ranieri's lab in Siena, Italy, and they're working on genetics, gene therapy, and COVID-specific research, and also Mamita Barua's lab in Canada. And they'll be joining us again in July to update on her lab's research, where they're trying to improve diagnosis by looking at the relationship between FSGS and Alport syndrome. I think I'd like to pass over to Katie now to just round us off before I conclude the workshop.
Thank you, Patrick. Firstly, from a patient's perspective, I just wanted to say that was an amazing presentation, very inspiring, and actually I think it gives a lot of hope to Alport patients. Personal view of that. I'd also like to thank you all, because I know you are currently all over the world right now. There is some people in England, some people in Italy, a lot of you are in the USA. So thank you for collaborating together to bring it together tonight. I'd also just like to say thank you on behalf of all of us to Susie and the Alport team, who works ridiculous hours behind the scenes, and this would not go ahead without her. So yes, thank you everybody who attended tonight, all the attendees and the panelists, and thank you to you, Patrick, for doing an amazing job, as always.
Oh, you're very, very
Thank
kind.
you.
I wouldn't be able to do it without Richard and Tomohiko, who obviously make this workshop look way smarter than I ever could. We will get a recording of this talk. If you missed anything or want to go over it again, the talk will be up on our YouTube channel at Alport Workshops in a few days' time. Otherwise, thank you all so much for watching over the last hour and a half, and have a lovely rest of the day or evening wherever you are.